Again, my name is Richardson. I am a early career data scientist here at the lab. You can talk to me about deer, tigers, or my dog. I love all of those things, but when I'm working, I'm usually working on different omics data with Lisa and her team. So today I'm going to be talking to you guys a little bit about the pre-processing of transcriptomics. So what is pre-processing? It's a very ambiguous term, and you're definitely going to get different answers depending on who you ask. So what we're going to call it today is right after all those steps that Bill talked about earlier, we have this tabulated spreadsheet essentially, right? So on our Y, our rows, we have all these genes and transcripts. I'm probably going to be using those interchangeably as we go through our presentation today, so bear with me. And then across the top, as our columns, we have different samples. Within this matrix, we've got different counts. So these are all integers, and we're going to be using this frame of reference when we're working through our pre-processing today. So it's going to be everything from getting this spreadsheet up to the point of we're doing the differential expression. And as a reminder, differential expression is our comparison between the two or more experimental groups that we're interested in. Okay, so because the term is so vague, what does pre-processing actually mean? So when I think about pre-processing, a lot of what I'm thinking about is quality control, first and foremost. So as Bill was saying earlier, there's a lot of things that can happen in the lab that maybe you're not anticipating, right? A lot of different things can happen. We're all human, and that's okay. But we need to find what has actually happened. So by doing pre-processing steps, we're doing data exploration, we're looking at our data. And if something looks strange, you're trying to find out why does it look strange instead of just taking it out because it's not performing quite as well as everything else. So that's a big part of pre-processing. Secondly, we also want to talk about the variation in our data. Naturally, when you're doing data analysis, you're going to get variation that is not related to the question you want to answer. For example, if you're looking at folks who have a tumor versus folks who don't have a tumor, the variation between two folks with a tumor is going to be less informative for you than the variation between folks with a tumor and folks who do not have a tumor. In order to isolate that variation, typically we do different processes of transformation or normalization to kind of reduce the variation we don't care about as much. Batch correction also falls into that category. We're not going to cover that today, but just something to be aware of moving forward. Lastly, we also want just an overall better understanding of the data. You guys know from Bill's presentation, there is a lot going on in transcriptomics, right? So understanding the trends, using some of those insights, knowing the biological background of your data can help you visualize in the correct way. Earlier, we had a question about bias and amplification. One of the biases that you can talk about with transcriptomics is actually GC bias, or the amount of GC in your transcript will influence how much of the machinery is actually going to that particular transcript, right? So this plot here on the bottom, let's see, it's on my right side of the screen. That is actually a little graph kind of displaying what that GC content looks like in your libraries. And that's really important to consider, especially if you're looking at downstream analyses, if you're looking at pathways. If something is extremely upregulated, maybe that's one of the things you'd like to consider. Like, is there different GC content in certain transcripts than others that we're looking at? Okay, so that's basically why. I'm sure there's a lot more reasons, but that's what we're going to cover today. So I'm sure you guys have been seeing throughout the week, people have done these steps. You've seen proteomics, you've seen other examples where they go through this process of trying to clean and make their data shiny before they actually do their statistical analysis. There are some special things about transcriptomics that we need to consider when we're doing this pre-processing step. So firstly, it's count data. Most data that we're working with and other senses are more normally distributed potentially than transcriptomics data. So count data is typically modeled by a negative binomial or a Poisson distribution. That are some of the graphs on our upper left. You'll notice that with different parameters, these distributions kind of change in shape. However, when we're looking at count data, we're going to see mostly that right skew. They're going to be hugging that y-axis that we're looking at. So we're going to get a lot of transcripts that have zeros, ones, very low expression compared to the very high expression transcripts. So if we think about what count data has in it, there are zeros. If you guys have ever taken a log of a zero in a program like R, it gives a very unhelpful value like negative infinity that doesn't really tell us anything. So that definitely comes into account when we're thinking about the different algorithms we can actually use on this kind of data. A second consideration that, is possible when you're looking at your data, not all of your samples will necessarily create the same transcript profile, right? You're going to get different expression levels across your different samples. Sometimes it's real, sometimes it's not. One thing we talk about is library size. If I have sample A and I'm seeing a total of half the reads as I see in sample B, but the genes are also expressing with that double, are they actually different? Typically, we're going to say that's not really the case. We honestly really try to adjust for that by taking the library size into account when we're doing our different processes, right? So again, big takeaway is that we really need to consider where we're coming from when we're working with the transcriptomics data and what we can actually do with integer values, these counts. Something to think about as we're moving forward. What other things besides a log transformation might zeros mess up, right? We're going to be talking about it a little bit more. So I'm going to walk you guys through like my process when I work with this kind of data from a, data scientist perspective. How do we go about the data? What do we look at? It's not going to be comprehensive of all the things you can do in the interest of time, but it's a good thing to look at and kind of get a sense for as we're working through this. The data we'll be working with today is very clean, very curated. It's from the developers of a package that is very popular for differential expression, EdgeR. This data set does not have 5 replicates, unfortunately, but like Bill said, it is very ideal for that to happen. Usually we don't get real world samples that are as controlled as these ones. So in this data set, we've got 6 mice. All of them are female. They're all going through the process of lactation development. We've got virgin mice, we've got pregnant mice, and we've got actively lactating mice, right? Additionally, we've got samples from different stem cell types. We've got the basal stem cell, which is kind of the background of what's your body doing while going through the process. And then we've got cells that are in the mammary gland, which are much more related to that developmental process. So based on this experimental design, you'll have a couple different ways to look at your data and do those comparisons. Additionally, just for notes, all these mice, again, are female and are genetically identical. It is a very controlled condition. Using Illumina sequencing, we are doing bulk RNA-seq on these different cell types. So let's see what it looks like. So as we see, we've got a histogram here and a box plot. Maybe you can't even see the box plot because this is non-scaled data. So the histogram shows us that there are lots of counts, as we were saying, kind of hugging that y-axis. We've got so many transcripts that are zeros or ones or barely, even there. On the other plot, again, we see the box plots are actually hugging the x-axis. Everything's very skewed towards 0. We do still see some points way up there, and those are typically related to your housekeeping genes. Like Bill was saying, there's some stuff that's happening in your body all the time. The housekeeping genes are going to be constantly transcribed, and they are helpful. They're very helpful. However, they might not be as interesting as seeing the full variation data source. So next question is, can we actually scale it? Can we do something to look at these plots in a little bit more informative way? So it's a little bit of a yes and no question just because it depends on where you're going. When I'm doing my processing, there are 3 specific packages, 3 specific differential expression workflows that I tend to use, and those rely on raw counts. So when we're going into that workflow, we are going to need our raw counts, not any transformation. However, when we are visualizing our data, we are exploring our data and trying to figure out what's going on, we absolutely should be doing this scaling method. Some examples of plots that are going to meet this are box plots, obviously, as you saw, some bar plots if you decide to go that route, principal component analysis, dimensionality reduction. We're going to talk a little bit more about that later, but those plots tend to expect normalized values. So you have to make very specific considerations for your transcriptomic data when you're going through those plots. Lastly, we've got some mean variance plots, which are very popular, but they essentially look like scatter plots in this slide. We're looking at mean expression values across these different samples. Okay, so we can scale. How do we do that? Like I said earlier, there are zeros. If you guys remember, logs of zeros are not great. The different methods that we can use to transform our data are, based on what you're trying to do. Again, to Bill's point, there's a couple different transformations you can do based on your gene size. So that's your reads per kilobase transcript per million, as well as fragments per kilobase of transcript per million. Difference between those is if you have single or paired end sequences, right? And again, if you've got a bigger gene compared to a teeny tiny gene over here, it's going to be more likely to get hit with that transcriptomic machinery than the small gene. You're naturally going to get a larger amount of reads from that. That is definitely a great consideration when you're looking at your. Typically, when we're going through the popular pipelines, though, we are looking at log counts per million or something very similar to that, because it's a pretty easy transformation that we're doing there. So how do we do this? We need to slightly elevate our zeros for this process to actually work with the log. What edgeR does is essentially it adds this 2 divided by your library size for a sample. So that's, again, the total counts of reads that you have in a sample. Counts per million calculation is pretty straightforward. You have the counts that you see in a transcript or a gene. You divide that by the library size, which again is the total counts within the sample, and that is your counts per million. And after adding this small increment, just log to that. That's what edgeR is going to be using when you're doing that pipeline. Limavoom does something very similar. However, it decides to add that increment in the counts per million stage as opposed to the log counts per million stage. It adds half a count to your numerator, which is your, for a particular gene, the number of transcripts that you see. And it will add 1 count to your denominator, library size. What this effectively does is make sure that no matter how many genes that you see in your sample, you're always going to get a number between 0 and 1. Just to let you guys know, that is not all of what's available. edgeR uses some very specific algorithms that are a little bit more involved. A regularized log, which essentially elevates, again, those small numbers, but does the same thing as a log transformation does at high numbers. And variance stabilizing transformation, which is also a very similar elevation that we have there. A note for these methods, the small numbers that they're using are kind of arbitrary, and they're just trying to slightly elevate those zeros so we actually can do the log transformation. All right, so lots of methods available to us. We're going to focus on log counts per million as we move forward with our data. What does this look like? As you guys can see here, we've got a couple different plots. On our top left is just what we saw in the histogram, but we have this log counts per million applied. You obviously can't see very much of the actual counts. You see a very strong spike still around 0. And that's, kind of expected based on what we were looking at in our histogram. If you actually scale the y-axis, you can see we're getting a little bit of a hump at right about 5 log counts per million. So that's a little bit more of a distribution we're kind of going for in the middle there. You can kind of see this reflected as well in our box plots. We're no longer hugging the x-axis. We're actually seeing a little bit of elevation, but that first quantile is still just right on that line. So different scaling can help us explore data in different ways, and we're going to keep looking back at these plots as we're going through, see it at the end and what we look like after we do the rest of our processing steps. So after doing our initial transformation, what I like to do is kind of start looking for problems a little bit. So once again, we're doing science. There's a lot of things that can happen. I'm going to present to you guys a couple different scenarios, and we're going to think about how we go and check and see if these are happening or not. This is by no means comprehensive. There are so many things that can happen in science in so many different ways that you can test for these things. So we're just going to go through these few today in interest of time. I also would like to point out that curated data is not the same as cherry-picked data. Just because it looks good, that doesn't mean you keep those and throw out everything else. It's very important for us when things like this, that we go talk to our people, talk to our analysts, maybe even do more plots, look at the specific gene, try to see what's going on before we actually remove it from our data set. Data is valuable, but you got to have reasons if you're going to remove some. Okay, so in the first scenario, oh man, I didn't even talk about the scenarios. We've got a couple scenarios here. So, like I mentioned earlier, lots of problems can happen. Enzymes are a big part of any transcriptomic process. And if your enzymes for some reason are left out on the bench too long, if, I don't know, you even hold it in your hand too long, it's too warmed up, maybe they don't efficiently transcribe your different samples. So how do we check if that is the case? We're going to take a look at some of the reads and see if that will tell us. A second example, if you guys have watched Breaking Bad, Walt was super focused on a fly that was in the room, and for good reason. If a fly gets into a sample, it can cause all sorts of havoc. So we're going to see what we can do to detect that by looking at all the different transcripts that are actually coming off the sample, all the non-zero transcripts. Lastly, this happens all the time. Labels get mixed up. It's very easy for a 13 to look like a B. You know, it's just something that's part of the process, but we have to know how to tell. So we're going to look at some dimensionality reduction to pull that apart. All right, now we move on. So this is our first example. We've had a denatured enzyme. There's something going on. This plot here on the left is a good example, right? Everything's about consistent as far as library size goes. On the right, though, we see one sample that is way lower than everything else. This is when you'd want to go back to your analyst and kind of ask, what's going on with this sample? Maybe do some more plots. Some of the plots that we'll show later on might also reflect some of these differences that you see here, but it's one of the first signs that maybe something has happened in your sample prep. Additionally, some notes when looking at these plots. This is a count plot. You won't need a transformation for this one, but it's also very important to look at your library size. Even if we're all looking consistent across the different samples, if you have low reads across all of your samples, that is also indicative of a problem. Additionally, there is a chance that this might be flagged earlier by sequencing software that it comes with, typically the instrumentation. So talk with your analyst, see if there's anything going on there. I know for Illumina sequencing, like 20 million reads, I think, is probably one of their baselines that they use for a good sample. So definitely talk with the folks who are actually running the process. They will know a little bit more about the data, probably. All right, moving right along. This plot looks very similar to the last plot. We're looking at the fly situation, though, right now. So here on the left, we have the good one. The right, we have the bad one. On the left, we kind of see very similar profiles as far as how many biomolecules in your sample are actually returning expression, returning counts. So comparatively, if you have one sample, maybe two samples that have a lot more transcripts that they seem to have that have expression, that might be a sign that something's in there that's not in the other samples. This is not to be confused with if you see one particular sample group. In those cases, you might actually see that true expression elevated for those samples. But overall, if you see something like this, that might be worth checking out. The kind of things you can do, again, if you know it's a fly, you go back and compare those transcripts against the expected reference for fly and see if that's where your alignment's happening. If it's for something else, it might be harder to detect, but again, talk to your analyst, see what might have happened. Great. So again, we also want to check our y-axes. If you're seeing very low transcripts across your samples, and this is going to pair with your library size, right? There's also a problem there. It's very possible to have a huge library size but only see a small number of samples. Like Bill was saying earlier, ribosomal RNA is extremely common in your cells, and it can potentially blow a lot of things out of the water if you're not careful with how you're doing. All right. Our next example is going to be the switch to labels, right? So this is principal component analysis, and I'm going to talk to it a little bit. So this is one of the methods of dimensionality reduction. So basically the idea behind it is that variation in your samples should be a little bit more similar across your groups. So if you can look at this plot here, we've got the reds all kind of clustering together and the blues all kind of clustering together. And the hope would be that the variation structure in your samples would be similar based on these. So this looks good here on the left, right? Here on the right, we actually see a little bit of color mismatching, right? The shapes are the same, but the colors are not. What this might tell you is that the variation does not look quite as it should for those samples. And then this is exactly when you go back to your analyst and say, hey, maybe something happened. What's going on? Additionally, these plots are extremely helpful. If we look at the plot on our left here, this distance between the points actually means something, right? So this distance is going to reflect that amount of variation. And we can see that there's actually a lot more distance between the blue and the red than there are between the shapes, especially when we're looking at the blue. We've got really strong differences between those different developmental stages, and the blue is the cells that are specific to those mammary glands. That's kind of what we expect, right? We're seeing a lot more changes in that cell as compared to the basal cells, which see a much smaller change between these different. So this can be one of the things you're looking at when you're thinking about your expectations of what your sample should look like. A note on PCA, you could have a whole lecture just on PCA. You could have multiple lectures on PCA. There's so many different methods that's been around for a long time. But typically, PCA is going to require normalized data, which is difficult with count data. How we do this here is a specialized PCA. This is a GLM PCA that can handle zeros. But in other methods, that's not necessarily the case. And you would have to use those transformations on your data before using PCA. There are other PCA methods that I've listed here. PCA Hubert, which is essentially robust to outliers in your PCA method. GLM PCA is what we have here. Once again, sparse PCA will try to essentially, when it's weighing the different variable components across your sample, it's going to essentially try to make it as small of weightings as possible for some of them and really emphasize the weightings for others. There's definitely a better explanation online, probably for sparse PCA, but just to let you guys know, lots of methods exist, and it might take some looking to find the one that's right for your data. All right, now what, right? We've done all this, we've gone through our scenarios. We've decided that, hey, our data looks okay. We've done a transformation step, we've checked it out. We've done a little bit of data exploration. And again, it's not comprehensive. There's much more things you can do, like that GC plot that we showed earlier. And we also, if we did see an outlier, if we could justify it, we would probably filter that out at this step. However, are we ready for the next step? Normalization, differential expression. If you guys have been around all week, you've probably seen that process in our other methods. The answer is no, we're not. But that is because we need to think about our statistical analysis that we will be running a little bit more. So again, we're going to be using the very common statistical differential methods, and that is DEC2, limma-voom, edge-R, and we're going to talk a little bit about what those assumptions are. I'm going to go over this a little bit more in our next talk, but I'm going to introduce you to it just briefly. So all of these assume counts, talked about that earlier. DEC2 and edge-R are going to assume a negative binomial distribution and assume that most genes are not differentially expressed. So that is a very important point. By default, it's going to assume that. Limma-voom uses a linear weighted model. The statistics are approximately normally distributed after you do your statistical analysis. And all of these are going to rely very heavily on ratios and distribution. We've been talking a lot about zeros. Zeros, ones, very low counts are really going to potentially mess up some of those distributions. If you think about it, if you see a gene in sample A that has 2 counts compared to sample B, where it has 4 counts, do we really believe that it's twice as expressed in sample B to A, or that we're seeing just very low levels of expression? It might be. Typically, it's the latter. So when we're doing these different processes, it's recommended that we do some filtering before we move into our differential expression. Additionally, we talked about this briefly in our workshop yesterday. If you've got a gene that you only see expressed once, that's not going to be incredibly helpful. So we also do a filter to take some of those genes out. We're actually going to do that one first. We like to look at a plot like this because it's stepwise. Again, it's discrete values when we're looking at observation. So what it would look like if we took out everything once for this data set is we would lose about 1,000 genes. That is not going to be the case for a lot of other data sets. It's usually going to be a much bigger number. For example, meta transcriptomic data set, which I have looked at recently, you lose about 40,000 genes, right? There are big data sets that exist. And this is an important graph to look at when you're deciding to choose your threshold. If you decide to keep all the genes that have observations across samples, like that can be a very robust thing to do. You can also visualize how many you're going to actually have left, right? And think about if that actually matters to you when you're thinking about those zeros and when you're taking out. Great. So there's also, again, looking at those expression levels, looking at those very lowly expressed samples. So on the right-hand side here, we have a good plot. On the left-hand side, we have a bad plot. But what are these plots? So these plots have the log counts per million on the x-axis and a density of what's going on in your sample on the y-axis. And this is kind of reflective of some of those histograms, once again, that we were seeing earlier. So if we have a giant spike at the beginning, way near our y-axis, it's not quite going to be what we want to fit our model to. What is recommended by DEC and EdgeR process pipelines are to use about 10 to 15 counts observed in at least some of your samples. And that is a function that we can get into later in the workshop. Or you can do a total sum across all of your samples and set a threshold there. Sometimes you can do both. It can be very helpful. But by applying these filters, you'll essentially set that peak at the beginning, come down, and you'll get more of that actual distribution that you're hoping to model. Okay. Now what? Now what does the data look like? So the top is what we were seeing before. We had our box plots hugging the x-axis. We couldn't really see that kind of distribution around 5 in the log counts per million. After we do our filtering, our box plots look like normal box plots. We can actually see the end of them. And we actually have a little bit of a distribution on the log counts per million around 5. So this is looking good, right? Are we ready? We have done transformation visualization. We've done some box plots, some data exploration, trying to make sure that everything looks correct for our data. We talked a little bit about our models, what we have expectations when we're looking at our data, to be able to fit the assumptions of the model. What's nice about some of these different analyses is that they kind of do the normalization step for us. So we talked a little bit earlier about library counts, right? They essentially will adjust for that for you. There is additional plot that I don't have a slide for here today that looks at that assumption that all genes are mostly, so rephrasing that, most genes are not differentially expressed. The way they do that in a plotting visualization sense is you can look at the ratio for a sample of a gene and compare it to the mean across the rest of your sample. If that ratio is about 1-to-1, that's what you'd expect for most of your genes. However, if you look at all your genes and you're seeing those ratios systematically above 1, that's kind of a problem. That overexpression is probably maybe something to do with your setup. So they do normalization factors that adjust for that. Lisa will be talking more about that later. But likewise, if everything was under 1, they'd bump it up just a little bit. So that is important for thinking about what's going to be happening to your samples as they're going through these differential expression processes. Again, a couple other caveats being batch effects. We didn't cover that today, but it's very important. Dimensionality reduction methods such as PCA can help you identify those. If you have batch effects, it should be considered. But again, for sake of time and ease of presentation, we're not going to be talking about this right this second. Additionally, as you're going through your pre-processing steps, as you guys could see, those box plots are changing. Those histograms are changing. it is very important to go back and look at what you're doing, your overall data structure, as you're doing these different filtering steps, especially if you find outliers, especially if you want to remove contaminants, right? What does that do to the data overall? Very important to know.